Good afternoon, everyone, and thank you for joining us for today's webinar. My name is Josh Vickers with RTI International's Forensic Technology Center of Excellence, and I will be your moderator and host for today's webinar. Today's webinar is entitled Variability in Blowfly Development, Research and Implications for PMI Practice. We do have two fantastic subject matter experts and presenters today. Our first we will introduce is Dr. Leon Higley. He received his bachelor's in chemistry from Cornell University in 1980, his master's in entomology in 1984, and then his PhD in entomology, crop production, and physiology from Iowa State University in 1988. His research focuses on insect ecology across basic and applied areas. His active work includes forensic science, decompositional ecology and blowfly physiological ecology, conservation biology, plant-insect interactions, and pest management theory. He is also a member of AAFS and the Entomological Society of America. Our second presenter of the day is Dr. Amanda Rowe. She is an assistant professor of biology at College of St. Mary in Omaha, Nebraska. She received both her PhD and master's degree from the University of Nebraska in Lincoln, where her research focused on blowfly maggot development and decomposition ecology. She also received her bachelor's from the University of Wyoming in 2006. Our quick disclaimer: the opinions, findings, and conclusions or recommendations expressed in this presentation are those of the presenter and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. And now, without any further ado, I would like to go ahead and switch everyone's layout and hand the presentation over to our presenters, Dr. Leon Higley and Dr. Amanda Rowe.
Well, hi, everybody. This is Leon. I'm in Amanda's office in Omaha. Amanda's sitting right by me.
Hello, everybody.
So we have the presentation that we'll make on the research. We'll just start off with a little bit of background. And I'm hoping this takes about 30 or 40 minutes, if I'm not too verbose. Amanda will keep a check on me. And then just open things up for questions. I do know that Amanda has a 2 o'clock commitment, so if she has to leave, you'll still be stuck with me. And I want to encourage you that if there's things we can't answer during this session, please don't hesitate to send me an email or even give me a phone call. Let me give you my cell number right now, which is 402. What is my cell number? 402. God, 402. I can't remember what it is, Amanda. Hey, we should go to the next slide while Amanda looks up what my phone number is. God dang it.
It's 402-560-6684.
Yeah. Good God. And my email is easy to find, but it's just LHIGLEY1@unl.edu. So the work I'm going to talk to you about is from a grant that I had with NIJ. Amanda was the principal investigator on that. As you're about to see, she did kind of a heroic study. And the focus of this is really on postmortem interval. And I think most of you who are in forensic science will know that in many cases involving, typically homicides, but cases involving where time of death is a crucial factor, it emerges that there aren't that many ways, particularly when you're looking at a body that's been subject to substantial decomposition. You're limited in the tools for trying to estimate that postmortem interval. And that's historically why insects have been used. And I think it's a fair statement that using insects remain the most accurate estimator of PMI, certainly when you're talking anything more than a few hours after death. Oh, I'm doing it. There we go. One of the problems with the PMI, and this is actually why I got involved in this work now 18 years ago, is that I personally don't believe the research data support our estimates of the insects models has been adequate. I say that coming from a lot of work in the ag background, and I was kind of horrified to see that we had much better data sets and better information for most agricultural insects in terms of estimating their development than has been true in forensic science. And I wish I could say this is a problem that's been solved, but it's not. But part of our intention here was to get a very robust data set for the most common blowflies, and then know that we would have solid estimates from that, and we could use it to develop more modern tools and improve the quality of our estimates, also to give us some measure of variability. So really specifically, that's what our goal was to develop a data set so that we would have better estimates for calculating PMI. And then, without getting into it too much, historically the way for estimating development has really been how fast you grow on one side versus how long it takes, and fitting a line to that. But as you're going to see from our actual data, that's not the way insects grow. It takes a long time at slow temperatures, then they have a rapid time through middle range, then they slow down again. It's more like an S shape. So when you see me use, or see us use this term, curvilinear models, that's what we're talking about. The other thing that complicates this is there's not one number. The factors that affect how fast you grow, they differ whether you're an egg, whether you're a larva, what stage you are as a larva, and so on. So just as background, and that's what we'll talk about here in the next few slides, when we look at decomposition of animals, probably the two biggest factors are temperature and oxygen. Temperature is important because it drives chemical reactions, but more importantly, when an animal decomposes, decomposition in the early stages really means being fed on. It's whether you're being fed on by another vertebrate animal, by insects, by microbes, fungi, and all of that's temperature dependent. If any of you have ever been involved in recovering bodies that have been submerged or in another area where they've been oxygen deprived, or in fact, if you've been on a case and you've lifted a body and smelt the underside of the body, you already know that decomposition is oxygen dependent, not unlike, what do I want to say, rotting potatoes. If there's oxygen, aerobic bacteria work, and that stinks. But if there's no oxygen, then you get anaerobic bacteria. It takes a lot longer, and they make the aerobic smell like perfume by comparison because they really stink. So we have these biological agents, all of which are temperature dependent, also depend on oxygen. And then we also have, as I mentioned, vertebrate scavengers, a very important factor when you're looking, say, with human bodies. If it's the right season and the body's available, you'll see insect scavengers. And let me say available, as you probably know, is everything from outdoors to in a house to locked in a trunk, pretty much anything unless you've got it in a freezer or somehow vacuum sealed. Insects can get to it. So to look at a normal time course in the summer, here's some work from research we did over 8 days. So each of the slides I'm going to show you is 2 days apart. So as we can see, this is actually, if I remember correctly, 20 minutes after death. We picked up these pigs who'd been killed with a humane killer. They're on this frame because we recorded weights through time. The cords are temperature sensors. But it received a killing blow right behind the ear. You can see that within 15 minutes there are flies, and these are blowflies, which will be a lot of the subject we're talking about. So there it is on the first day. 2 days later, we can see that there's a, we can see, let's see if I can do it. Here we go. You see here, there's a maggot mass here, and you can see that the nose has lost a great deal of tissue. We already can see that fluids are coming out of the body. These fluids, sometimes called stinker juices or whatever, they're very high nitrogen, and that's why you see the blackening. It's actually the same thing as if you over-fertilize your lawn. It's killing the grass around here. And as we look through time, we can expect to see that halo of death around the pig. So let's continue. Here we can see that there's complete skeletonization of the head. an incredible maggot mass. We have full bloat in the rest of the body, but notice back here, these feet look like they're barely decomposed, whereas this skull is already starting to be skeletonized. This is a normal pattern of animal decomposition going from the head down because the flies have to lay eggs at openings in the body. It's also one of the reasons that I tend to be a skeptic. I don't know how Amanda feels about this, but I tend to be very skeptical about rating systems people devise for decomposition because it's not a single thing. This part of the animal is not decomposed relative to this. And so trying to suggest that there's a single stage when you can see everything from skeletonization to really what looks like something fresh, I think is misleading.
It's also very subjective because you're judging decomposition based on what you can see on the surface. And a lot of times you'll have decomposition happening underneath and you'll have the skin covering the top, and it's not until you lift up the skin that you can see the decomposition that's happening underneath. And so it really is based completely on the person who's scoring it and their past experiences with decomposition. So they're not super useful when you're estimating rate of decomposition or what stage a body is in based on observations from the outside.
And I think another proof of that is you keep seeing these things crop up every 3 or 4 years in the scientific literature. Somebody comes up with a new system, and then on testing it really doesn't work that well. One other thing I want to say relative to the point Amanda made is what we're looking for is something that's dead reliable, where it's not going to be influenced subjectively. And the thing I can say about these insects is that while there are some subjective considerations that we can talk about, their growth is a function of the temperature and time. And so really our subjective things are when were the eggs laid, and then we might say, well, okay, were they exposed to sun or whatever, what was the temperature? We may have questions about the temperature when we try to go back in time. But in terms of the process of their growing, we should be able to determine that with a great deal of scientific accuracy. And that's really the point of the research we did and are doing in this presentation. Okay, if we go a little bit further forward, I keep forgetting that, we can see then that the body's deflated, the maggots have moved here to the center. We would actually, if we were going into this in more detail, we'd see additional insects at other parts of the body and so forth. Now, this brings me to the next thing. I want to make an important distinction between when you have insects and don't have insects. And we have some slides to illustrate that point as well. There are 2 pigs, both killed within a couple minutes of each other, both set out at the same time. You'll see the one on the right here has there's a shadow. That's actually a shadow from a cage. We put cages over both of these, but this cage had openings. One cage had openings so the flies could get in. The other cage, we tried to keep the flies out as best we could. So this was on the 1st of June, a long time ago that doesn't feel so long. And this is 1 week later. And so it illustrates kind of what the role is of insects. And this could also be true if we're talking about, if we're excluding vultures or other vertebrate predators. A lot of the tissue that's removed is removed by what other animal or whatever creature, I should say, can get to it. If you block access by scavengers, be they vertebrate scavengers or insects, well then all you're left with are bacteria and fungi, and that is, they may have fast growth rates, but in terms of their ability to convert tissue, that takes a long time. And that's also, of course, why you see the bloat here from those gases that are essentially respiratory gases from the bacteria.
And most of those bacteria are anaerobic bacteria. So as soon as that skin opens up and oxygen goes inside, all of those bacteria will die. All of those gut bacteria that are flourishing right now, all of them will die. And you're basically just left with a mass of tissue that doesn't go anywhere. So it really is the insects or vertebrate scavengers that reduce the biomass when we're talking about decomposition. Otherwise, the bacteria and fungi take it to about this stage that you can see on the screen, and then as soon as oxygen comes in, they're done.
And we use pigs here for these field studies. The process is no different for a decomposing human. And I might also say, if you're like some of the students that Amanda and I have had, if you're curious when you have that big bloat about what happens when it ends, let me just say that don't do what one of my students did, which was to kick it with his tennis shoes And yes, they do explode, and decompositional gases and fluids pour all over. And as I like to think, that stain's not coming out. So there are a variety of species. Our original intent was to look at 5 species. Unfortunately, we were only able to look at 2 given the scope of what we had to do. Actually, what our plan was, we had hoped we'd find shortcuts after we did the first species. And what we found was not only could we not find shortcuts, but things were much more variable and we had to do extra effort. So instead of looking at 5, we were only able to look at 2 over the course of our study. But we picked what we regarded as the 2 most common species associated with bodies in North America, Lucilia serracata and Phormia regina. Now, I'll have Amanda talk to you about just briefly go through the details of the experiment. You don't really need to know what we're about to show you on the details, but there is an important point that we want you to take away from this. And the point is, well, I guess the point is going to be, what does it take to have an accurate understanding of development? So here, by way of background, let me just screen through this. And I'm not going to take credit. I think Amanda put these together. They're really good. So the blowflies belong to this insect family, the Calophoridae. They're typically the first flies at a body. They're really adapted for finding carrion rapidly. There's different types of fly species that can come, and in some cases we'll find other things, but what, 90% of the time I would say it's going to be blowflies. So all of the blowflies, and indeed all of these higher flies, non-mosquito-like flies if you will, have the same essential life cycle. They start with an egg. The egg hatches and you get the first stage maggot, and it has some increase in size. Very shortly after it comes, you get the second stage maggot. Again, some increase in size. But let me warn you, size is not a good measure for growth because diet can influence this. And then finally, there's a third stage larva. And as you can see, this is a big larva that probably 90% of the feeding actually comes from this. And then after it's done feeding, it doesn't do what any normal animal should do, which would be like a butterfly, spin a cocoon, and well, oh no, it wanders off the body typically. They're much bigger, it wanders around, and eventually it finds a place to form the pupal stage. It might be a little hard to believe if you're not an entomologist, but this structure, the pupa, or what entomologists call the puparium, is actually this. It's the outside skin of the last larva. When it forms the pupal stage, it actually pulls away. This thing tans, compresses into what we typically call a barrel-shaped, mahogany-colored, or pick your own color. And then inside it, eventually the adult fly will form, which I think is next slide. We don't have it here, which is the only flaw in this slide, but it gets out of here. There's a weakened seam on the pupa, and I mention this because a fluid-filled balloon pops out of its head called the tulinum and pops this off. And to me, it's one of the most peculiar features in all of nature that to get out of a sleeping bag, you blow a balloon out of your head. Where did that come from? But anyway, that's how they do it. So here's what our goal is. Our goal is to try to get a very comprehensive, detailed estimate of how they grow at different temperatures. And we don't just want to measure how long it takes for them to grow to one temperature, but we want to get some measure of that variability. So we don't want to look at a single time. We want to look at multiple times when we think they're going to molt, so that in the end, we can have an estimate for every stage: the egg, the first stage larva, second stage larva, third stage larva, the third stage larva when it's wandering around, what's called migratory, and then how long the pupal stage lasts. So Amanda, you want to walk us through this quickly?
Sure. So Leon mentioned earlier that we originally wanted to do 5 species, and because the data collection was so intense, we only actually completed 2. But for both of those species, both Lucilia serracata and Phormia regina, we looked at 11 temperatures. So we started at 7.5 degrees Celsius, we went to 32.5 degrees Celsius, and we moved in that 2.5 degree intervals. So 7.5 degrees Celsius is low 40s,
40s,
upper 30s Fahrenheit, which is really at the tail end of their development range. So we didn't really have hardly any development at 7.5. For Formia regina, I don't think we had any development. And then 32.5 degrees Celsius is getting pretty warm, so we're up in the 90s at 32.5 degrees. And that's, again, when Leon mentioned that when we talk about curvilinear, we're talking about this S-shaped curve. We have slow development where it's cold. We have really fast development when it's hot until you reach a certain stage, and then they just start dying. They start cooking. So 32.5 degrees Celsius is reaching their upper range. They don't really go past 40, at least these two species don't. We looked at a 16-8 light-dark cycle, so the lights were on for 16 hours. They were off for 8. Previous research indicated that light-dark cycles can impact development, so they can speed it up or slow it down depending.
And just to interject, this is one of the reasons why you will hear forensic entomologists argue over which data set is right or wrong, because lots of the old data sets were done under constant light. And what research has subsequently shown is that alters the development rates. So sometimes the variation we see between what one researcher has done is because of the experimental technique. So what you would hope to do is try to identify those features, remove them, so that you're getting something that's much more realistic.
We had, for each temperature, we had 4 replications. So each temperature had 4 separate chambers with the same number of containers in them. We looked at 6 stages, as Leon mentioned earlier. We went from egg to adult eclosion, meaning they used their telinum to pop out of their pupal case. We had 5 subsamples per stage transition. So for each one of those individual stages, let's say egg, for example, we sampled within that stage 5 times. So we started at the very beginning when the eggs were first laid, and then we sampled 5 times within that stage until we got to L1. We sampled 5 times within that stage, so on and so forth until they all emerged as adults. We had 20 eggs per experimental unit, and we had 120 experimental units per temperature. So those 120 experimental units are what we divided between those 4 chambers. So you can see that here, the cup on the top that says E3, that was our experimental unit. So there's pine shavings in there, and there's a little cup that held the 20 eggs. For Formia regina, we used sand because we found that the Formia regina maggots, once they hatched out of the egg and they were in first stage, they are really tiny and we couldn't find them. We tore the liver apart. We're using beef liver inside of the cups for them to grow, for them to eat and grow on. And we couldn't find them. They were disappearing. And what was happening is that we found out that Formia regina has an interesting larval behavior where they wander. So they wander off of the liver, and what they were doing was they were actually, if I can get this little arrow, they were coming out of the cup and they were circling around underneath that bottom lip, which is why we couldn't find them, because they were under the lip and they're really tiny and they're fairly translucent, so they're easy to see through. In the pine shavings, they were falling off of the cup into the pine shavings and they were desiccating or drying out and they were all dying. And so we put the cups down into moist sand so that they would crawl out as first stage larvae. They would crawl around, but then they had a way to get back into the cup onto the liver so they could continue their life cycle. Each of those boxes was 7 by 7 centimeters, and they were 10 centimeters high. They had 2.5 centimeters of wood shavings or 2.5 centimeters of sand. We had 29.5 milliliter cups. Those are those little cups on the inside, and each one had 10 grams of beef liver.
The beef liver issue matters because we want to make sure there's enough food so that there's no starvation. That doesn't affect development. But at the same time, we know if we have too much food, bacteria will move in and it can contaminate, and that will confound things. And again, this is another thing that when you look at the scientific literature, sometimes people focus on what they fed the maggots, sometimes they don't. But it's another explanation for why we're seeing variability in people's results. And just to illustrate where we're going with this.
So this is what, on the left-hand side of your screen, this is what an egg mass for blowflies looks like. There's probably 20,000 eggs in that mass on that piece of beef liver. So we would put fresh beef liver into the cages. So we have cages of adults, mixed sexes, mixed ages, and we would check every 15 minutes, and as soon as when they started laying eggs, that's when we started our time. And they had half an hour from the time they started laying until we got the mass size we needed. And if they didn't hit the size we needed within 30 minutes, we had to start the experiment all over. So we started it the next day. We then counted those eggs out into 20 egg groups. You can see those circled in the blue outline there. And then each of those 20 egg groups got transferred to one of those small cups from the previous slide. And then they each got put into their own individual box, and then that box got put into the chamber.
It's every bit as difficult as it looks. I would say Amanda became a master at sorting and counting these eggs. But you'll notice that when the flies lay their eggs, they actually lay them in these groups, and there's sort of a sticky, I guess you would call it glue. I suppose it's a mucousy substance that helps keep the eggs together. Clearly in nature, it's to the advantage of the fly for all these eggs to hatch, and by feeding in a group, they do better on a substrate than if they were there singly. But it was a nightmare counting all of these eggs. But in terms of the experiment, it's just one nightmare among many.
Yep.
I don't know what did I do here.
Okay, so these are what our chambers looked like. So those 4 chambers on the left were just looking at a single temperature. We randomized the experimental units, or those little clear boxes within each chamber, because we wanted to make sure that we weren't influencing the development of the maggots based on which shelf they were on or how close to the light they were. So in each chamber, they were completely randomized inside. On the right-hand side, you can see what it looked like to have those experimental units inside of the chamber. This was after we had gone through most of the life stages. So you can see here that there are migratory, that's what the M stands for, and pupal experimental units that still had not been sampled yet. So we were waiting to sample those.
I see there's a question about how we determine the right amount of meat. The answer is that this is the part you don't see. Why do experiments? It seems so straightforward. Why does it take you years to do the experiment? And that's exactly the reason. We did a whole series of preliminary experiments to try to establish the proper ratio of meat to immatures. Another concern we had was whether having 20 eggs, if the feeding behavior was different than if we had 200 eggs. And so we tried to experiment to make certain that we weren't somehow getting a bias in our results, that maybe if, for example, if it was a lot easier for 100 to feed, well, then maybe their feeding rates would be faster, more typical of what you'd see, versus 20. And so the way we figured that out was to...
we just did a series of smaller experiments, and we wanted the number of maggots to be large enough that they would feed, because if you have too few maggots, sometimes they just die. They don't develop. And if you have too many, you also have to take into account as maggot numbers increase, they form what we call maggot mass. And the larger the mass, the warmer the temperature is. And so they start developing within their own kind of microenvironment, and they can maintain the temperature even if the ambient or outside temperature is lower. And so we didn't want maggot mass temperatures to influence the development of the individual maggot.
And again, just to interject, this business of maggot mass is its own big problem, because people usually, you can take measurements, say, oh, they're hot, they must be reproducing there. Well, actually, they're too hot. They're right at lethal temperatures. Development rates we know are slowed then, plus they're doing a lot of moving to try to prevent themselves from literally from cooking, and so they're not feeding there, and so it still remains an open question about how fast the insects develop in the maggot mass because of these various factors. So we knew that was beyond what we could do here. We wanted to get the baseline, and then, and in fact, excuse me, we are even now working on maggot mass issues about oxygen tolerance, because it's a low oxygen environment, some other issues. But I would say of more interest to nerdy entomologists right now than to your average forensic scientist.
And we did have some liver left. We did that on purpose so that the food wasn't the limiting factor for their development. Maggots, if they run out of food, will sometimes pupate earlier than they would otherwise. So there was a little bit of liver left, but we didn't want a whole bunch of liver left because then we're promoting the growth of bacteria and fungi. So.
And the other thing I guess we should share with you, the reason we're telling you all about this and the reason we spent a year or more just trying to get this was really a response to the scientific literature. We kept looking at papers and finding what we regarded as deficiencies in their methods. And we kept seeing results that didn't match what we'd seen in nature. We got in arguments with people at scientific meetings. We still have arguments with those people. And we decided that rather than allow opinion, the right thing to do was to do the best, most precise study you possibly can. And once you have that, you've got a basis then for trying to look at other studies. And if there's disagreements, you try to determine whether the disagreement is because of how the thing was done or whether it's really reflective of some biological difference. I wish I could say our work had resolved that, but people are pig-headed. That's what I'll say about it. I guess I'm on record now.
To give you an idea of just the sheer amount of work, for the total experiment, we had 20 eggs per experimental unit, 120 experimental units per temperature, 11 temperatures in 2 species, for a total of over 52,000 eggs. And that doesn't include the time that we had them all counted out and then had to stop the experiment because the chamber broke down, or we didn't get enough eggs or something like that. So in actuality, we looked at hundreds of thousands of individual eggs. And then if we look at it overall for sampling times, with 2 species, we're looking at 6 stages within that species, over 11 temperatures, and 5 samples per stage. There was over 660 sampling times. And one of the reasons that this research hadn't been done to this extent before is that very thing. Maggots don't sleep. They don't stop eating. It's a 24-hour-a-day, 7-day-a-week sampling schedule until they emerge as adults, until their life cycle is complete. And so you were in the lab 3 a.m., 4:30 a.m., 2 o'clock in the afternoon, and when you're running multiple temperatures at the same time. So we were lucky enough with our NIJ grant to be able to buy multiple chamber sets, so I could run 3 or 4 different temperatures at once, and I would try to stagger the sampling times as much as I could, but you're in the lab all the time. I mean, You're constantly there.
For about 2 years, Amanda had, well, probably permanent sleep deprivation. I mean, she was in the lab pretty much all the time, and she makes a good point. There's nothing technically difficult about the study we did, but practically it's nearly impossible. And this is another issue that began to bother us when we looked at the scientific literature. People report development times, but they don't do what we do and complain about how hard it was to get those times. And nobody could do an experiment like this and not come away complaining about losing their whole life in terms of measuring development. So what that really means is that people are probably measuring development on a daily basis. Maybe they come in on weekends or whatever. And so if somebody says that, oh, it took 5 days, well, did it really take 5 days, or did it take almost 6 days, or did it take 7 days? And that becomes a very big issue, particularly when at some of the higher temperatures where there's rapid development. So I always use the word heroic with Amanda's work here because I don't know how else to characterize it. It was very, very demanding. Okay, let's talk about the fun part and what we found. The first thing that Amanda came is, and I think you guys are going to be excited when you learn that apolysis and ecdysis didn't represent a single molting event. So I don't see the catch, the question and answer coming up with wow and so forth. So what does that even mean? Amanda, you want to do this?
Yeah. So when maggots go from one stage to the other, they molt their outer skin just like a snake does when it grows too big. Their skin doesn't grow with them. They have to develop new skin and shed their old skin. And so apolysis is what we call in entomology that actual change. So in that picture on the right, you can see where that maggot has undergone apolysis, where it's growing its new skin has grown underneath.
Yeah, so this is the old skin, the transparent part, but the white and everything below that's old skin, and these 3 lines are associated with the new skin. The old skin has these 2 lines.
Right, so that's how we tell maggot age. We look at the number, those are called sporacular slits, and those are what the maggots actually breathe through, one of the ways that they gas exchange. And so this picture is actually a 2nd stage maggot becoming a 3rd stage maggot. So it's changed, but it hasn't molted yet, and then we call that ecdysis. So it hasn't shed its old skin yet. So you can see the 2 slits in the sporacular plates on the 2nd stage maggot, and then the 3 slits in the 3rd stage maggot underneath. And this was something that had not been clarified in literature before, this kind of relationship between ecdysis and apolysis. It was just assumed, and that happens a lot, especially in forensic entomology. There's a lot of assumptions that people just run with instead of actually looking at them and testing them. It was assumed that they followed very closely together. So as soon as the maggot changed, it molted and shed into its new age, and we found that to not be true.
And what's one of the things that's significant about the graph, and this is something you'll hear us talk about a lot. Recall, we had the eggs start at the same time. We counted eggs, tried to get them on the liver within no more than an hour of when they were laid. It was kind of a frantic thing. But notice that even though they're under constant conditions, blah, 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 that we see variation. As you can see right here, on average, we could see that there were some that were 2nd stage, some that were the start of this sort of cryptic 3rd stage, and some that are really 3rd stage. We had a mixture of these things, and that mixture lasted, what are we looking at here, Amanda? 10 hours, more than half a day.
Yeah.
Probably between 24 and 12 hours. So you'll hear me say this again, but often when you attend workshops or even read books on forensic entomology, you'll hear people talk about collecting the oldest maggot. That's absolutely wrong. You should collect a representative sample of the maggots, because if you just collect this and think you're here, or worse, here, you've already made a big error in your estimate of the PMI. And you're going to see us repeat this observation with other data that we have. So one of the things we're interested, of course, is in how long they stay in each stage. That's what we call, and how long it takes to go from one stage to another, the point I was just making. And as you can see, this can be a substantial period of time. This shocked us. It's not that all of the maggots jump from one stage to another in a couple hours. It can be days while we'll still see this mix of stages. And that's something people have largely ignored in talking about variation. So the fact that the transition to go from one larval stage to another, from larvae to pupae, takes such a long time was completely unexpected to us. And we can say it's not because we had a highly genetically diverse population or anything of that nature. And it's also not because of an environmental factor, because we were maintaining the larvae under very strict, uniform conditions in these tests. What does it mean? It means that there's a lot of variation in there naturally. And I think that fits with work that other people have seen, that within populations there's genetic variation. It probably is kind of it's a spreading the risk thing. It's the same argument about why you don't want to put all your retirement in a single stock. And so for these maggots, if they spread out their development, if some arrive too early and get killed, well, there's others that will go later. It probably increases the average likelihood that they're all going to survive. But it was unexpected. We don't know why that's there, so we're going to keep going. So, key findings. For our species, we didn't find them growing below 7.5 C.
They would hatch. The eggs would hatch, but they wouldn't get past 1st stage. They would die.
A big thing you'll hear me talking about a little bit more detail in a moment is that when they go from stage to stage, they follow a bell curve. That's important because people had, and a lot of the recommendations for forensic science seem to imply that, no, it's not a bell curve. They all emerge, and then there's a long tail. That's not true. That means that trying to, again, this is another example of why selecting the largest ones for picking maggots is not the right, they're not the right way to do it. In response to Donald's question about how to do this, we're working on it, but there's a pretty easy thing to say. Every place on a body where there's maggots, you need a sample, and just go ahead and take a spoonful right out of the middle. And I guess if you're going to err on one side, take too many, not too few. But don't be distracted and bias your sample by trying to just get hunting around for the biggest maggot. What's really true for all kinds of sampling, whether it's insects or if you're on a factory line sampling, you want to take things randomly, and then you base your estimate of where you're at on research that's also based on random samples. What we want as forensic entomologists is just an honest sort of, I didn't bias this sample, with enough individuals so that it's representative of what you see. And so my tool of choice had long been a plastic spoon I stole or was contributed from McDonald's, and I'd take a couple spoonfuls to put in a vial from each of the sites.
So if there's 10,000 maggots, don't just collect 10, collect 300. It's better to have way too many than too few, and it gives you a much better idea of the transitions that are happening and what part of development that maggot group is in as a cohort. So it's not necessarily, we don't have a rule of thumb. Just collect more than you think you will need.
Yeah, that's exactly right.
From multiple areas.
So I'm sure this graph may not mean a lot to you, but basically it's showing graphically the transition from one stage to another. And in this instance, you can see a couple of things. First, it's not the same at each temperature. You can see that they kind of go up the S. So where these two lines cross, that means there's half of the one stage and half of another. You might say, well, Leon, if I got you a really good sample from a body and it was 70% of one and 30% of another, could that tell you how long the body had been there? In principle, yes, it could. The big problem are these friggin' error bars, and you'll notice that at high temperatures, they get huge. So there's much more variability at high temperature, and certain stages, as you can see here, have higher variability, all of which tell us that we need those large sample sizes so we're not making a mistake. And as an entomologist, it means I want to be cautious in any PMI I make when I'm talking about, as you can see, if I'm here where I'm not seeing any that are in the pupal stage, there's less variability than if I'm here with the big transition between the pupal stage. And so this actually helps prevent a guide for an expert to say, okay, where can I be? How cautious do I need to be relative to my conclusions? Another thing that we were able to do, which I thought was pretty cool, is that we could do this thing called probit analysis. It's a technique that's used usually for figuring out responses to poisons. But what it let us do was model the actual shape of these transitions mathematically, so we could use them in a predictive way, looking at what these percents are. But you'll notice some of these lines look really weird. Well, they're really weird here because Sericata at 10 degrees C is not happy. A lot of the individuals are just barely hanging on. And then you'll also notice that it's for certain stages, you can't really see them here, but you will in an upcoming slide. It just doesn't look like things work as properly as you might like. So a lot of these graphs I'm showing you now with the stage transitions were a way for us to meet that goal of how do we reflect variability in our estimates for time of death. By looking at not just the oldest stage, but at the distribution of stages, we can actually look at that variability. These dotted lines represent the 95% confidence interval. They say that we would, with 95% certainty, we should see such and such a percentage and a stage here. And that's what each of these curves allow us to do.
Crystal, the survival rates for the samples at extreme temperatures are not good. So thinking back, at 7.5 degrees, we had none get past first stage. They all died at first stage. At 10 degrees so this is for Phormio regina, let's see, I'm trying to remember off the top of my head. We didn't make it past, again, first stage at 10 degrees. It wasn't until 12.5 in both Formia and Sericata that we started to see some survival, and it was dependent also on the experimental unit. So we tried to get the best eggs that we could, and you can kind of tell a bad egg from a good egg by color and inflation. So if it's nice and plump and kind of cream colored, that was a good egg. Bad eggs were gray and deflated looking. But just because the egg looked really good doesn't mean it was going to hatch. So that was just a natural mortality rate inside the species. So some of our survival, I mean, it was hard to measure survival just based on temperature, other than at 7.5 and 10 degrees, we had nothing past first stage. At the warmer temperatures, we didn't see, at 32.5, that's not quite warm enough to start to see die-off from it being too hot, but we did some outside sampling at 35 and 37.5, and that's when we started to see a lot of die-off. So between 50 and 75% is what we were seeing at those really hot temperatures.
And that just is an interesting observation. We can go in the field and measure maggot masses with Formia or Sericata, and the center temperature might be over 40. And so that's one of the reasons that I can say with some confidence, even not having even done the experiments, that those individuals who are in those masses are not really experiencing that temperature for any length of time. Their movement, I don't know if you've ever seen a mass of maggots, but the crawling and writhing, I'm becoming increasingly confident that what you're seeing is they're trying to cool off, that when they're in the middle of the mass, it's so hot they're going to die, so they have to constantly move around. And that affects feeding rate. They're using energy, so that's going to slow their growth. And it's not going to be an easy question to resolve that because of the complexity of it. But I guess what I do want to say is if someone tells you, oh, I used blank temperature to represent a maggot mass and they're going to grow really fast or whatever, that's simply not true. And it's not supported theoretically. It's not supported from what we see. With respect to the humidity, the answer is yes and no. Typically, in a very dry environment, it can be problematic. The eggs can desiccate. They'll lose water and just dry out, which is undoubtedly one of the reasons that the flies lay their eggs at openings where it's moist. Experimentally, as long as there's enough humidity, they'll go ahead and feed and so forth. It's really more of an issue that if it's extremely dry and that dryness isn't offset by local humidity from the body, then you can get mortality or will affect growth. So it's not so much, it's more like a switch, on or off. It's a fair way to say it, isn't it, Amanda, than it is that there's a response to humidity. They don't grow with response to humidity the same way they grow with response to temperature. And I would say probably the most important feature with respect to with this issue of humidity is that when we look at the high desert or basin range area in the West, those conditions are so drying that finding a body, say, without maggots or with very few maggots is not as surprising, just because if there's a high wind speed, if it's a murder in the day, it may become so dry that that window of opportunity for flies to successfully lay eggs may close. So there's not been as much research done under those conditions, and it's an area where I think you have to have a lot of caution. I know that's how we've approached cases we've had.
Yeah, plus you have the drying out of the body fairly quickly. So if the fly manages to lay the eggs and the eggs survive and hatch into first stage, the tissue from the body itself might become too dry too quickly for them to complete their life cycle all the way through to adult. So they might just run out of food if that tissue gets too dry.
Lou, you asked a question about accounting for different opposition of different flies. If you'll bear with me, right at the very end of the presentation, I can answer that question. It wasn't part of our study, but it's an illustration of what our research has led to. So if I don't answer it, bring your question back up, but I think we can answer it here at the end. Okay. Remember I said that we would like to model things with curves rather than lines because that's how they grow? This slide actually illustrates that. The one thing that's kind of peculiar is we see here with Sericata, just as we'd expect, really long and then it goes down, really long and then it goes down. And then here, for what's going on here for those migratory? That's not the right, where's the curve? When you see this, where it's the same time across all temperatures, what that tells us is that development or that behavior, in this case, the wandering behavior, it's not temperature dependent. They wander for a certain amount of time, and then after they're done wandering, they go ahead and form the pupal stage. And I think what's peculiar about that is that why are we the first people to have noted it? I mean, it's a pretty important feature of their biology, and yet I don't think anybody recorded the fact that this is temperature independent. And I think that too is a sign that the data sets and the actual data people have used really have not been as detailed as they should have been. That was very surprising. It was a surprising finding to me. Okay, I'm going to go through this quickly because we haven't bored you already, this will definitely do it. You can actually use these degree days to make use of it for forensic purposes. We plot the reciprocal of, we plot 1 over days in stage. So this is essentially fast development going up, and then temperature, and we fit a line to it. So that's what we did here. Did it for both species. We have our 95% confidence intervals, which is cool. I'd like to point out we got a lot of points on this line, and it's statistically valid in that each point is separated by the same amount of x-axis difference, which is another flaw we saw in the literature. And what do we do with it? Well, we use it to calculate degree days. I don't know if you've seen a graph like this, but you probably, those of you who've ever worked for this know about there being some minimum developmental temperature, and then you take the mean temperature and you subtract one from the other, and this area represents the amount of growth. And if that doesn't really ring a bell with you, then you're very fortunate to not have gone through it. We made a spreadsheet of this stuff where we look at, here's how long it took to go through the stages. We ran a regression, and from that, we figure out something called ADD, which is accumulated degree days. We have the range for which this equation is valid, the range in temperatures, and it's not all the same. And then we also look at what % deviation there is, a measure of variability. And so this is where we actually come up with the number we can actually use in calculations. And you'll notice that we have different numbers of degree days, and we have different values for our developmental minimum based upon the stage. Recall I said it wasn't a single number, and we can take care of that now. Whoops.
But what we want to see is the mean from the data looks similar to the regression accumulated degree days that we got from the models.
Yeah,
so that's what we're looking at here, making sure that the information from the data matches the modeling that we were predicting.
Right. So this was like an internal test on what we were doing. A second internal test we did, if the assumption for a degree day is valid, then we should get, when we look at that, we should just get flat lines, and that's pretty much what you see. We did get flat lines. In other words, that, yeah, we've got the development rate, and it's valid across these temperatures, and for temperatures it's not valid, well then we have to exclude it. One of the things that we did, there's been probably the most widely used data set is one from Kamal in 1958. And to be honest with you, it's not even a regular data set because it's only a point. So even though it's been used for calculating degree days, that point, you only have one point. So how do you even do the calculation? Well, you have to assume that the other point should be 0 or something else. So what we did was, we've heard arguments that Kamal had very accurate data for the points he looked at. So we calculated degree days by his method, and we compared that to what we calculated. And what we were surprised to find is how close, with just a couple exceptions, we were. This one exception is, recall we said they're not temperature dependent. This stage is just a time dependent thing. So the fact that these vary doesn't surprise me at all. There was quite a bit of variation here in the third log. Oh, sorry. I was using my cursor. So these vary because the migratory stage isn't temperature dependent. These vary because it's a long stage. We'd expect to see a lot. But The surprising thing to us was how close they were, which implies to us that Kamal's data, sort of going ahead with what people had argued, looked pretty darn good. And ironically, we didn't want to get into a fighting match with people, but we have the tables hidden in our laboratory where we compared all the other papers, and they don't agree. And often we can say, well, I think they don't agree because this method was right or this method was wrong. But by seeing a study that before we started we thought was really good, match our data closely, was encouraging. It also had some implications that I don't think people like, which is that we're skeptical about reports regarding geographic variation, because in those reports we never see a really strong discussion of variation the way we did in this study. So we have the suspicion that people are confusing local variation because they're not doing enough replications and so forth with geographic variation. And the fact that we have similar results from Kamal in Washington to our work in Nebraska using flies that were originally from West Virginia, I think we tend to interpret that as supporting our skepticism. So we're almost at the end, at least of this part. Just a couple of things to finish. So we got our procedures well established, and if anyone else were brave enough to do this for other species, I think the stuff we worked out are great. The stage transition information I think is extremely important. It definitely influences how we want to sample and how we need to get proper, what the data are so we can get proper estimates. And I think that fits to the next point. And then often you'll see at the end of any study, oh, there's new, this is going to lead to new research or so forth. Let me show you an example, and this is actually what contributes to the question on oviposition. There's been a long-standing argument that the reason the blowflies, there's so many different blowflies that come to, and other flies that come to a body, is that they have evolved for different times and so forth. But to the best of my knowledge, nobody's ever looked at that. So Amber McInnes, a student who just finished her master's work with me, picked up on some of what Amanda did. We know the development rate. So she looked. We actually did a competition study. I'm not going to go into a lot of details, but this is replacement series experiments are an ecological way to study competition. What matters is we vary the proportion of two flies. We put them on diet at the same time. If they were equal competitors, where these two things would intersect would be exactly at the middle. And what we discover is, depending on, and here we compared our same two species, Phormia and Serracata, and what we discovered is that's not the case. And the reason for that has to do with development rates, which is tied to oviposition. It turns out that if certain flies lay their eggs faster than others, they can grow up to a point where they will be able to effectively compete with another species that, if they arrived at the same time, would outcompete them. And one of the things we were interested in is that because the hairy maggot blowfly is now in the United States and it's been spreading, how does it compete with native species? And what we find is there's lots of evidence of that competition, and it's very much the way it's mediated is probably through when the flies lay their eggs. So I would expect to find that as this fly becomes more common, I'll bet we start seeing behavioral switches in some of the other species to lay eggs earlier than have been previously the case, because those that lay eggs later are going to be outcompeted and killed. So really what you're doing is it's selecting for it's likely to select for flies that will oviposit earlier. And we can also use these data to predict what kind of flies we're likely to see on bodies.
It also includes, so there's some species that develop faster. So Rufifaces and another non-native blowfly called Megacephala, they both came into the United States. Well, they came through Brazil in the early 80s, and they made their way up into the United States. Rufifaces will now make its way up into Canada, I believe. We have some, I know they come in to Nebraska at the end of August, very beginning of September. And then we don't see them anymore. They can't survive the cold temperatures here. Megacephala barely makes it out of Florida, and Megacephala, for any of you guys that are in the South, especially in Florida, Megacephala develops faster than these other species, our native species. And so they are right now the dominant species. Even if Saricata and Megacephala laid their eggs at the same time, because Megacephala develops through its early stages faster, it outcompetes Saricata or Phormia.
Sam, I see you asked the question about how our results might differ if instead of using constant temperatures, if we had used fluctuating temperatures. It's interesting you posed that question because I made a vow with my students that before I die, which I think I better get on it because I'm facing my third cancer here, but I want to resolve that issue. It's kind of a technical issue in terms of measuring development. I can say very briefly that I know that our data are still valid for use in the field because, as I'm about to show you, we've had some tests. What I think we're going to find is it will shift where we are on the development curve slightly, depending on how severe that daily fluctuation is. The reason it's complicated is because there's a mathematical issue in looking at fluctuating temperatures called the rate summation effect. I only mention that in case you want to look at it. Sometimes that's called the Kaufman effect. And then there's also biochemical questions. And really what the fluctuating temperature thing relates to is the fact that, as weird as this sounds, we kind of don't know at a biochemical level how temperature affects development. That seems like a really big thing to say, but the details of that are still kind of uncertain. Now, you could talk to lots of other scientists and they'll say what I said is wrong, but I'll challenge them to show me the papers that definitively show that process. And I think when you start looking, you suddenly realize, yeah, we know chemically what it does to reactions, but that's not the same thing. So, so the short answer is there's enough. Here's what I think the short answer is. Our constant temperature estimates are good enough for what we see in the field. There's so many other sources of error. The error that's associated with not looking at fluctuating temperatures is washed out. But on an absolute level, we would want to look at how that changes. And really what we want to do is relate constant temperature measurements to the fluctuating temperature measurements. What I will say is, if you have temperatures that say go from 0 to 10, and I've made measurements at 5, that's not going to be the same. A constant temperature is not going to be whatever the average of those fluctuating temperatures is. At low temperatures, they'll grow faster than predicted. At high temperatures, they'll grow slower than predicted. But whether that's for biological reasons or just because of mathematical reasons based on the shape of the curve is still kind of an open question.
And again, we were just looking for a baseline. We knew that if we started with a constant temperature, it would give us the baseline we needed so that we could start incorporating some of those other aspects that could impact development. And so we just haven't made it to the cycling temperature part yet.
But yeah, but we're getting there. And it's kind of the same thing as what we did with the competition.
Yep.
We have used our data on a variety of cases. We listed some of them here. Amanda and I do some casework together, but we're primarily researchers. So although I don't know, we had been doing a couple, maybe anywhere from 2 to 3 cases a year. I think that's going to increase dramatically. We had worked with Neil Haskell, I think is the premier forensic entomologist. As Neil's health has faded, he's asked us to do more cases, and so I think that will probably change our caseload. What I can tell you is that in at least some of the homicides, the Joplin, Missouri homicide, the myiasis case, that's infestation of it wasn't a criminal case, it was civil. It's infestation by maggots. In both of those cases, there were other lines of evidence on the postmortem interval, and what we found lined up with those other sorts of evidence. I think the really profound test will be if I can get access to Neil Haskell's data set. Neil has done something like 1,500 cases. He's getting close to 1,500. And of those 1,500, I believe there are 100 cases where there's either confession or there's confirming evidence about time of death. So really what you would like to do is to look at those cases, run our model, and see how accurate and inaccurate we are against those cases. And I'm hoping that as Neil puts his database together, or we work with him putting the database, that will be a better test. But in terms of using it, I guess I'm going to make the same thing with respect to our own data I make with others. We try to be extremely conservative in our estimates. I tell people that if somebody's telling you to the hour or half day when the murder was done, they did it, because we don't have that kind of accuracy with our data, barring certain unusual events. So I think it's important, particularly from the NIJ standpoint, for those of you who are actually working in homicide investigations, to know that what we've been able to develop is useful, and in my opinion, I think we're getting much more accurate results. I reanalyzed some of my old casework, and it was lining up better with what I've been told from police afterwards than my original analysis did. So, and I think that's it for us. So we can turn it over to you guys for any other questions, if you have those. Thanks so much for listening, and I hope this was informative and helpful for you. Amanda?
Yeah, and we are open to doing cases. So if you guys have, mostly we're just in the lab, but if you guys have evidence that's collected during a case, feel free to send it to us.
Or just need advice or whatever.
Yeah.
It's part of our, I think it's part of our civic duty. That's what we'll say.
All right, thank you so much, Dr. Higley and Dr. Rowe. If you wouldn't mind, I know that you had given out your email address earlier in the talk. If you wouldn't mind just typing that there in the chat pod on the bottom left of your screen, that way the audience members will be able to write that down before we do leave. Also, just for our audience members to know, we are recording this webinar as all of our other webinars, and we will be turning it around into our archival version over the next few days, and then we will send you out a reminder email that is ready. We are also, when we go through our closing here in the next few minutes, we do have a PDF version of today's slide deck that you will be able to download. So be sure to hang out just for a little bit longer so you will have access to that. While we are waiting on any last minute questions to come in, you both answered most of them during your talk. Do you have any final remarks that you'd like to give to our audience, just where your research is going or what they can expect next?
One thing I can say, it didn't come up in the introduction, but I have a big research project in Yellowstone looking at beetles that occur in the thermal areas, and what that's led to is a big focus on how insects tolerate high temperatures. So because of that, I'm pretty confident within the next year, my research group and probably Amanda and I will start looking more directly at the maggot mass temperature issue. I think it'll probably be longer before we look at the cycling temperature, just because we know how much work it is to do those studies. And that's not to say we won't do it, but I think what we're looking for is maybe a grad student with no home life or severe ADD where they're willing to live in the lab or whatever it is.
We do have, so we have our Sericata, the Lucilia sericata data is published. It's an open access journal called PeerJ, and I believe if you just go to peerj.com, peerj.com, and you search for our names, I think you can search by author name, and you can download our Sericata data directly for free. It goes into a lot of what we talked about today, as well as our actual data set. So we also published our Excel spreadsheet showing all of the data, which is not something that you see when you're normally looking at papers, but we wanted it out there so people could use it, they could test it, they could run it through their other models. We really want it to be as robust as possible. So yes, look through PeerJ for the Sericata publication. The Regina publication is coming. Life happened between 2014 and now.
Oh, the truth is just difficulty getting it published, getting past reviewers. There's a lot of contention within forensic entomology, and for whatever reason, people are not willing to readily accommodate views that are opposite. And I think there are a lot of people who do not like what we found, and that's okay as long as you can tell us we did something wrong. But if it's just a matter of opinion, I don't know what to say about that. There was a question about preventing the wandering stage from escaping the box. That's a good one. The larger box that cups in is sealed so they can't get out. So what's important is the box needed to be large enough so that they could just keep wandering that time when they wander, and then feel comfortable either getting into the sand or the soil. And that again was an experimental thing. If it's too small, then it affects the behavior, and it could have been much larger, but that was Amanda. Yeah.
Yeah. Well, then we wouldn't have had enough experimental units, and the boxes, those outer plastic boxes, also had a hole in the top with a mesh covering to let oxygen in and gases out, so we weren't smothering them slowly. The experimental temp, 32 degrees Fahrenheit. We went higher than 67. 32.5 degrees Celsius is, I believe, in the mid-80s. Let's see, 26.
I'd use my phone if it weren't a hot spot right now.
And we didn't document barometric pressure or humidity. We did just make sure that there was some humidity. So it kind of floated around 60%. We would test it every week, once a week. We'd look test the chambers just to make sure that we had some humidity, but we didn't monitor it every day.
Yeah, Jim, if you look at studies of humidity, it's a bear to both establish it and to maintain it. And so usually what's done is people will have like an open container water in a chamber so that they get a saturating humidity, say over 70 or something like that. If you're going to try to look at precise levels of humidity, then you use salt solutions and you monitor those. And we didn't think it was worth the effort because we don't see evidence to suggest that humidity is driving the development rate, other than what we talked about for those extremes. I don't think barometric pressure, it might have an impact on oviposition. Lots of insects measure barometric pressure in terms of flight behavior, those sorts of things. For larval development, it's kind of hard for me to see why they would care. And I say that because in the field, let's say you have a low pressure center with rain, you'll see the maggots go underneath the body. They kind of move with response to light and water, I think. At least that's been my supposition when I've looked at the stuff we've done in the field. It seems a little unlikely to me that they respond directly to those because they're not direct inputs into how they grow. I mean, the big input, they grow faster if it's warm, and they grow faster depending on how much they eat. Things after that tend to be, well, if it's too dry, I'm going to die, I have to do something, or if it's windy and I can't stay, you know, I'm drying out, now I have to get under the body, those sorts of issues.
The eggs were late. So we had in the lab, the average temperature in the room where we kept our eggs was a fairly constant temperature. It was inside, and we had fans. We had heating fans and AC fans depending on the season, so we could keep the temperature fairly constant. And it floated right around, I think, 25 degrees Celsius, which is like mid-70s, which was a comfortable temperature for them.
But Jeremiah, you asked a question that worries me.
Yeah.
Because in some insects, it's known that conditions that the female, and even worse, the grandmother, conditions that the mother or the grandmother are reared, actually influence the expression of genes in the offspring. So it's not out of the realm of possibility to say that could something happen in terms of the eggs and how fast they grow based on the condition the mothers experience. One of the reasons I think we were a bit relieved to see the agreement that we had with our data and the Kamal data is Kamal's data was actually done in a screen house outside. And so, although admittedly, at summer temperatures. So we didn't see anything to suggest that that's a phenomenon that occurred. And I don't know if it's, I don't think it's been documented. It might have been documented in Drosophila, fruit flies. I don't know if it's been documented, but it's a consideration. So Jim, you're wondering about extreme temperatures. between autumn and winter. You can find, if you're talking about finding live insects, it is possible to have, say, a spring discovery of a body that has a maggot mass on it that's still alive, and those would be from eggs that were laid in the fall. I know of cases where that's happened in Illinois. Neil Haskell would talk to me about cases he'd had where he'd seen that. Essentially what happens is they eat a little bit, enough to maintain their metabolism, and they're just sort of, I won't want to say they're hibernating, but they're just sort of in, they're not, it's not cold enough for them to die, but it's not warm enough for them to keep growing. So they eat enough to keep their metabolic heat, they sort of huddle together, and then as conditions get better, they will go ahead and emerge. When you're talking about really extreme temperatures, extreme highs and extreme lows, we don't see a big change when we look at field data when we have, let's say, daily temperatures we know are way above the lethal temperature. And undoubtedly what's going on there is that the maggot masses move around the body. And if it gets very, very hot, they will move in a place where it's cooler. More than likely that will delay their development. They spend energy doing the movement, they spend energy coming back and forth. We also know that their development rates slow at very high temperatures, just naturally. My guessing is going on is they're essentially turning on their physiological air conditioners. They're paying a lot of the food they're taking in that should be going for building fat, they're probably spending on building special proteins to help them resist the heat, that sort of thing. So I hope that answers your question. Hey, Amanda has to take off, so just to let you guys know, but I'll stick around and answer any questions.
It was wonderful talking to you guys, even if it was just virtually. So
It's encouraging to see.
Maurice, yes, we did. That's why we had so many experimental units, because one of the things in previous research was they would look at the maggots, or they would test the maggots out of the same experimental unit, and research has shown that disrupting the maggots like that or bringing them out of the chamber and putting them back can alter development rates. And so we did. That's why there were 120 experimental units per experiment or per temperature, was so that at each sampling time we destructively sampled. We took out one box from each of our chambers, so we took out 4 of our replicates at a single time, and we had enough experimental units to do that 5 times within each stage. But that's a really good question.
Yeah, and it also illustrates why we think that technique at looking at the same cohort has underestimated variation, because statistically it's called repeated measures, but what you're doing is you're measuring the same thing over and over at different times, and so that's not going to be reflective of the amount of variation that if you were looking in different individuals. So that, even though we knew that we were going to be adding a tremendous amount of work, that was another reason we did sample by experimental unit.
All right, thank you so much, Dr. Rowe, for your presentation and your talk today. We will give everyone just a couple of more moments to give any questions that they might have. While we are waiting on any last-minute questions, we'll just kind of move into our closing, but please feel free to keep typing there in the chat pod if you do have any questions. Again, we are recording today's webinar, and we will be converting that into our archival version here over the next few days. We will be sending out a follow-up email to everyone that registered. That way you will have the opportunity to either review back to today's presentation, or you can pass along to any colleagues that you feel would benefit from today's webinar. Again, we do also have in the bottom left-hand corner of your screen a PDF version of today's slide deck. Simply click on the title of the file, and then click the Download Files button directly below that, and you can download today's PDF presentation file before you do leave today's room. We do also have in the center of your screen our customer satisfaction survey. If you simply click on the word here, that will open up a new window for you where you can run through our quick survey that just gives us a great input from you, our audience, about different ways that we can not only change the content, but also change the way we disseminate that content out to you, our audience. So if you wouldn't mind taking a few moments to go through that, we would definitely appreciate that. Let's see, it looks like one more question came in for you, Dr. Higley, asking, were your time intervals for sampling per stage determined by treatment-free trial experiments?
Marie, yes, they were, and we also tried to look at what was in the literature. And so we ran, so basically what we wanted to do was we wanted every time the insect molted, we wanted to get the time we hit 50%, 2 times before, 2 times after. But of course we don't know that time because that's the whole point of our study. So we tried to look at, we did some preliminary experiments, try to see roughly what that was. Tried to make some estimates. For the most part, we had a really good spread. There's a few times when we missed it a little bit, and but on the whole, we were close. You know, one thing I've thought about with respect to trying to get other species and addressing this issue is that when we do cases, I would say more than 90% of the cases that I've worked on have involved third stage larvae, either third stage larvae, third stage migratory larvae, and pupae. And that's because the first and second stages are completed very rapidly. So I don't know if it's a cheat, but one thing we could consider doing for other species would be to just focus on those stages. And we do the same thing, right? We figure out the estimates. And then we would at least have data that would be solid for what's likely the most common stage collected. It's not as ideal as having every life stage, but I think probably there's going to have to be some compromises like that, because I just don't think many people are willing to undertake the kind of study that we did. And to be honest, I mean, it's expensive. We would not have been able to do this experiment at all without the contribution of the National Institute of Justice. Because of them, we were able to buy enough chambers so we could properly replicate it, and we had enough support for Amanda's PhD. It also provided support for Amanda's PhD, and you know, so there are practical barriers to doing these things, which I think should be. I do want to acknowledge that. I think that I'm happy we were able to do this. I thought it was important, and I'm also happy that the NIJ agreed when we did our grant application, because really without their support we never could have been successful. Man, geographic variation, I'm absolutely interested, Maurice. I would send me an email and we can talk about how we might want to proceed with that.
All right. It looks like that was the last question that has come in, so we're going to go ahead and wrap things up for today. Obviously, we would like to first thank Dr. Leon Higley and Dr. Amanda Rowe for their presentations and their research today. So definitely thank you for taking the time to present today. We would also like to thank, obviously, our audience for joining us today. Without your participation, these webinars are just not possible. So definitely thank you for your attendance and your support. Again, make sure to check out our website at www.forensiccoe.org for all of our no-cost forensic webinars that we do have up. We do also have our podcast there, and we are in our 5th season of our podcast, so make sure you check out the podcast as well. You'll see Janine put our website up there so you can have easy navigation to that. Again, thank you so much, Dr. Higley and Dr. Rowe, for your presentation, and thank you to our audience. We hope everyone has a great afternoon, and we can't wait to see you in our next webinar.
Josh, thanks so much.